All right, welcome everyone to the final lecture of the semester on lipid biosynthesis. So, we've already seen a number of examples involving reciprocal regulation of complementary cycles where you have something, say, like glycolysis, where you are consuming carbohydrates, and then gluconeogenesis, where you're producing carbohydrates. And you have these complementary systems based on both anabolic and catabolic counterparts, in that particular example, around glucose. Now we're going to look at the other half of lipid metabolism, which is the anabolic component, the biosynthetic component. The synthesis of fatty acids and other lipid components is inherently different in their regulation from that of the degradation products. So fatty acid synthesis involves a set of reactions that follow a strategy different in several important ways from the corresponding degradative process. Firstly, intermediates in fatty acid synthesis are linked covalently to sulfohydryl groups of proteins called acyl carrier proteins. Or ACPs. In contrast, fatty acid breakdown intermediates are bound to the sulfohydryl groups of coenzyme A. So we'll say versus coenzyme A sulfohydryls. So they have a different type of carrier which involves a sulfohydryl linkage. Secondly, fatty acid synthesis occurs in the cytosol, where fatty acid degradation, which we just discussed, takes place in mitochondria. So this is a cytosolic process versus mitochondrial. Thirdly, in animals, enzymes of fatty acid synthesis are components of one long peptide chain, fatty acid synthase. There's no similar association which exists for degradative enzymes, which are carried out in separate enzymes. So we're going to use a single specialized fatty polypeptide chain, fatty acid synthase. For lipid biosynthesis, at least the core of it. And then finally, fourth, coenzymes for oxidation reduction reactions in fatty acid synthesis are NADP plus and NADPH, whereas degradative processes involve NAD and NADH. So, because this is a synthetic process, and these are the four critical differences between our biosynthetic processes and our degradative processes. So, let's move along. How are fatty acids synthesized? Generally, we need to walk through a number of conditions before we can get to the overall cycle of synthesis of palmitic acid from acetyl-CoA. But make a little space here. So the big picture is that it's the formation of malonyl-CoA which activates acetates units for fatty acid synthesis. So fatty acid synthesis requires formation of malonyl-CoA, which is an activated state of our acetate units. So the design strategy generally looks like this. 1, fatty acid chains are constructed by the addition of 2 carbon units derived from acetyl-CoA. All right, now that we've erased that junk off the screen, that seems to just love to habitually pop up. We'll start over again. 1, fatty acid chains are constructed from the addition of 2 carbon units. And these 2 carbon units are derived from acetyl-CoA. So just like we break down lipids for energy into 2 carbon units, we are constructing all of our lipids from 2 carbon units as well. 2, these acetate units are activated by the formation of malonyl-CoA at the expense of ATP. And this costs energy in the form of ATP. Thirdly, the addition of 2 carbon units to the growing chain is driven by decarboxylation of malonyl-CoA. So like I discussed in class, whenever you have a decarboxylation, a minus CO2, you are creating a product which is highly stable, highly oxidized form of carbon, and this helps drive the reaction as an energetically favorable step overall. 4, the elongation reactions are repeated until the growing chain reaches 16 carbons in length, which is palmitic acid. My goodness. And then once we have palmitic acid, which is our 16 carbon fully saturated lipid, other enzymes then add double bonds and additional carbons. So that's the big picture. Now, let's elaborate a little bit. Remember, fatty acid synthesis depends on the reductive power of NADPH specifically. And the net reaction for the formation of palmitate from acetyl-CoA looks like this. We've got, whoa, dragging things around here. If you have an acetyl-CoA and 7 malonyl-CoA, which will be formed from acetyl-CoA, plus 14 NADPH, plus 13 protons and a water. This will yield 1 palmitate from bringing together the acetyl-CoA and the 7 decarboxylated malonyl-CoAs, 7 bicarbonates, 8 coenzyme A's, in the reduced form, and 14 NADP+. It's important to note that levels of free fatty acids are really low in typical cells, and the palmitate made in this process is rapidly converted to CoA esters in preparation for the formation of triacylglycerols and phospholipids. They don't linger. They are repackaged and repurposed quickly for biological function because free fatty acids need to be traffic carried, manipulated, and then processed quickly. Now, scroll up a little bit more here. It's important. Cells must provide cytosolic acetyl-CoA and reductive power for fatty acid synthesis. And this is actually quite a dilemma biologically to provide suitable amounts of substrates for fatty acid synthesis. It's not trivial to have enough acetyl-CoA, and it's not trivial to have enough reductive power in the form of NADPH. So, sufficient quantities of acetyl-CoA and malonyl-CoA and NADPH must be generated in the cytosol for fatty acid synthesis, and that's really the trick. Malonyl-CoA is made by carboxylation of acetyl-CoA, so the problem really distills down to generating sufficient acetyl-CoA and NADPH. So, lipid synthesis really requires cytosolic acetyl-CoA and NADPH. And that's the biological problem we have to solve. So there are let's start with acetyl-CoA, three principal sources of acetyl-CoA. Firstly, amino acid degradation. Can produce cytosolic acetyl-CoA. Secondly, fatty acid oxidation produces mitochondrial acetyl-CoA. Notice that's mitochondrial. There has to be solution to relocate it into the cytosol. And then thirdly, glycolysis yields cytosolic pyruvate, which, after transport into the mitochondria, is converted to acetyl-CoA by pyruvate dehydrogenase. So we can couple glycolysis yields pyruvate, which, after goes into the mitochondria, can be converted to acetyl-CoA by pyruvate dehydrogenase, but then it's in the mitochondria, and we need to get it into the cytosol. So the acetyl-CoA derived from amino acid degradation is normally insufficient for fatty acid synthesis. And the acetyl-CoA produced by pyruvate dehydrogenase and by fatty acid oxidation can't cross the mitochondrial membrane. So it can't participate directly in fatty acid synthesis. Instead, acetyl-CoA is linked with oxaloacetate to form citrate, which is then transported from the mitochondrial matrix to the cytosol. And here, it can be converted back into acetyl-CoA and oxaloacetate by ATP citrate lyase. So this cycle is called the citrate-malate pyruvate shuttle. And what it does is it provides cytosolic acetate units and some reducing equivalents for fatty acid synthesis. So this is really the solution for getting materials produced in the mitochondria into the cytosol. so that we can use it. I'm not going to have everyone memorize this cycle per se, but I'm going to zip through it real quickly. So let's say we have to get our inner mitochondrial membrane going on here, and we have our matrix side, and we have our cytosol. Please go away. And on the matrix side, if we have our oxaloacetate, this can feed into the TCA cycle and couple with acetyl-CoA, yielding citrate, which we've seen as the first step. And we can get this acetyl-CoA from fatty acid oxidation, primarily, as well as from other mechanisms. We could even get it from pyruvate, which has then been acted on by pyruvate dehydrogenase to form acetyl-CoA. And most important thing, we can take the acetyl-CoA, regardless of its source, from pyruvate or from fatty acid oxidation, couple it to oxaloacetate in TCA, and make citrate. And then the citrate is actively shuttled to the cytosol. The citrate can then be returned, oh my, to acetyl-CoA and oxaloacetate. I'll make a little bit of space here, give us some room. Oh no, I did it. Well, instead of trying to get rid of this thing, I'm just going to write over it. So it goes back to oxaloacetate, and then we can interact with malate dehydrogenase to make malate. And then we can have malate interact with malic enzyme to give us pyruvate. And then pyruvate can cross to the other side through active shuttling. And what's neat about this is that the steps, malate dehydrogenase needs to consume NADH, but malic enzyme produces an equivalent of NADPH. Also note, because this is taking place in the cytosol, glucose can produce pyruvate via glycolysis. So we have a couple different ways to get to pyruvate, to replenish pyruvate on the other side, and then this pyruvate can become, rewrite it, can go back to oxaloacetate via pyruvate decarboxylase. Also malate can actually be reshuttled back, and malate through malate dehydrogenase can go back to oxaloacetate, or pyruvate can just join up here and go to acetyl-CoA directly, once again through other mechanisms. So this is, in essence, how we're getting equivalents of acetyl-CoA from the matrix side to the cytosol. Is acetyl-CoA couples to oxaloacetate via TCA, making citrate, citrate shuttled, decoupled back into acetyl-CoA on the cytosolic side, and oxaloacetate, and then can be converted to malate and pyruvate, both of which can be actively transported back and reconverted and recycled again. Additionally, remember, glycolysis can contribute to pools of pyruvate, which can then be shuttled back into the mitochondrial matrix where it can regenerate equivalents of acetyl-CoA. So, pretty slick system for separating our reductive equivalents and keeping our anabolic and catabolic processes of lipid metabolism neatly separated. Now, another really important step in all of this is to recognize that there's a lot of acetyl-CoA in the cell on both sides of the mitochondria membrane. And because of this, it's important when we start lipid biosynthesis that the investment in this expensive energetic process isn't lost. We don't want the acetyl-CoA to suddenly find another fate metabolically once we've committed the energy to it. So acetate units are going to be committed to fatty acid synthesis by the formation of malonyl-CoA. Oh no. Apparently, try this again. Oh, look, can I erase it now? Nice. So this is a committing step synthetically. Once we've gotten to malonyl-CoA, that acetate unit is committed. It does not have another alternative fate. And this reaction is catalyzed by acetyl-CoA carboxylase, which contains a biotin prosthetic group. Okay, so I think we've set the stage at this point for looking at the pathway of palmitate synthesis from acetyl-CoA and malonyl-CoA. Let me make a little bit of space. And what we'll probably end up doing is zooming around a little bit once we get going here. But I'm going to do a cursory illustration of the pathway, and then we'll probably dig into it just a little bit of extra detail. But it's important to set up that this entire reaction is really initiated by, or to rephrase, the individual steps in elongation for fatty acid chain is really just a cycle of elongation that involves 6 enzyme activities. And it's initiated by the transfer of the acyl moiety of acetyl-CoA to the acyl carrier protein by a complex called malonyl-CoA, acetyl-CoA, ACP transacyclase, also called MAT. And it also transfers the malonyl group of malonyl-CoA to ACP. So what this MAT does is it's going to transfer the malonyl group and the acetyl group from malonyl-CoA and acetyl-CoA to acyl carrier protein. Remember that that's just a protein with a sulfohydryl group that replaces the coenzyme A from the acetyl-CoA and the malonyl-CoA. So when we see MAT interacting with malonyl-CoA and acetyl-CoA, this is the initiation of the synthesis of palmitic acid. So, that said, let's see if we can do this justice. We've got to erase that. So here we have our acetyl-CoA, and it's going to interact with MAT, our transacyclase, And that's going to move the acetyl group off of the coenzyme A and onto the acyl carrier protein. Through sulfur linkage to the ACP. Now, this is going to interact with the another enzyme, the beta-keto acyl synthase. But first, I need to set up the MAT interaction with malonyl-CoA. So let's see if I can do that over here. Let's see. Make a little bit of room. Get this all the way over here. See if we can do this. And here we have our malonyl-CoA. And notice it's just a carboxylated version of acetyl-CoA where we've added an equivalent of CO2 to the top of the acetyl-CoA. And what's going to happen is that we're going to have our acyl carrier protein with its sulfhydryl group come in and interact with the malonyl-CoA. And this is going to kick off the coenzyme A. And give us an equivalent of malonyl-CoA that is now coupled to the acyl carrier protein. This is being facilitated by that same transacyclase enzyme MAT that we saw in the first step. So now we have both malonyl-CoA and acetyl-CoA transferred to acyl carrier protein. Now enters the next enzyme, Ks, which stands for beta-ketoacyl ACP synthase. And it's going to be responsible for our initial link of the acetyl-CoA to the malonyl-CoA. Now, Ks initially interacts with our ACP-coupled acetyl group up here. You know what? I'm going to put little blocks around the enzymes for clarity. And what's going to happen is it's going to pick up our acetyl group from the ACP, and it's going to liberate that ACP Let me see how I can best draw this. And couple the acetyl group to the synthase enzyme. So now we've moved this equivalent of acetyl from the ACP to the synthase enzyme, and this simultaneously liberated our acyl carrier protein so that it could be used to couple to the malonyl-CoA, so we weren't using too many equivalents of ACP. Now what we have is we've got our ACP coupled malonyl, and we have our synthase coupled acetyl group, and via the action of Ks, we're going to couple those two. And this coupling reaction is going to be facilitated through the decarboxylation event, which I illustrated with minus CO2 here. And this is going to be our first synthetic step. And what this produces is an equivalent of acetoacetyl ACP. So now we have our, let's see here, CH3, our acetyl group. We have 2 carbons from our malonyl group. And everything's still bound to the sulfohydryl moiety of the acyl carrier protein. And this is acetoacetyl ACP. Make a little bit of room here. For those of you who were paying a lot of attention to the last lecture, you probably recognize that we now have a carbon which has been sandwiched by a pair of ketones. And this is going to cause an electron deficiency in this carbon, which will make it particularly subject to different reactions. Now, following the formation of the acetoacetyl ACP, we're going to bring in an equivalent of NADPH for its reductive power, and we're going to drive this reaction with another enzyme, beta-keto-acyl ACP reductase, which makes sense because we're bringing in all that reductive power. NADPH plus a proton goes to NADP plus, and this is going to be driven by Kr, which is, once again, beta-keto-acyl ACP reductase, R for reductase versus synthase in the first step. And what we're going to see here is the reduction of the first ketone drawn to an alcohol. So in a lot of ways, the opposite of what we saw in beta oxidation. And this is called beta-hydroxybutyryl ACP. Once we have our beta-hydroxybutyryl ACP, we're going to go through a dehydration event, which is going to give us a double bond. And this is going to be facilitated by another enzyme, beta-hydroxyacyl ACP dehydratase. Make a little bit more space here. DH for dehydratase, beta-hydroxyacyl ACP dehydratase. Let's draw this. We've now got our double bond. And now we have our crotonyl ACP as our product. From here, we're just going to go through another reduction with another equivalent of NADPH, and this is going to be facilitated by the final enzyme, beta-anoyl ACP reductase, or ER, for anoyl reductase, and get our NADPH plus our proton going to NADP plus. And what this is going to do is get our reduction of the double bond to a single bond. And this is going to give us our butyryl ACP. which is butyryl for butane, 4 carbon, 1, 2, 3, 4 carbon chain. Notice, I'm going to zoom in a little bit, shrink this up a little bit, make it a little, there we go. What we've done is we've started with our malonyl-CoA and our acetyl-CoA. Malonyl-CoA is just an acetyl-CoA which has been carboxylated with a 3rd carbon, and it will involve that initial decarboxylation in step 1 with the synthase enzyme. But we've gone from a pair of 2 carbon units, one of which was activated by the CO2. We're going to join them via decarboxylation into a 4 carbon chain which has a pair of ketones. We will then use an equivalent of reduction via NADPH and a reductase enzyme to reduce one of those ketones to an alcohol. We're then going to dehydrate, forming a double bond. We're then going to bring in another NADPH to further reduce that double bond to a saturated system, and that's going to give us our butyryl ACP. Now, what happens with the butyryl ACP is the following. What happens is this activated malonyl ACP, instead of bringing activated malonyl to the party with step 1 over here with the initial acetate or acetyl-CoA, what's going to happen is it's going to come right over here and interact with our butyryl ACP. And now what that's going to do is it's going to add 2 carbons to our butyryl ACP, exactly the same fashion as our acetyl ACP. Going through step 1 with KS, in fact, I'll even draw that in here. Repeating step 1 with the product that had been added onto. And we're going to go all the way through the cycle again, and then we'll have a 6 carbon chain. And then we're going to start over again, and then we'll have an 8 carbon chain. And we're going to repeat this process 2, 3, 4, 5, 6 times until we have palmitate as a product. And palmitate's a 16 carbon chain, so it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Palmitate. And that's our process for the synthesis of palmitic acid from acetyl-CoA and malonyl-CoA. So, let me make a little bit of space here. It's important to keep into perspective when you see a pathway that these concerted sequences of reactions often take place via a concerted effort between all of the enzymes that have been listed. And you carry out, specifically, build fatty acids on megasynthase complexes, which involve multiple copies of these enzymes organized three-dimensionally in a fashion around reaction chambers that facilitate the rapid conversion or production of palmitate. So in a mammalian fatty acid synthase complex, it looks, excuse my art, something like this, where we have our KS, or our beta-ketoacyl ACP synthase, tethered to our transacyclase. And then we have this reaction chamber, or this void, where the lipids combine. And then we have two equivalents of our beta-hydroxyacyl dehydratase. We have our KR, or our reductase. And we have our ER, our ennoyl ACP reductase. So we have our different reductases. We have two equivalents of the ER here. We have two equivalents of our KS here, lumped together. And this means that substrates can enter the reaction chambers and be passed from the MAT to ACP, to KS, to KR, to DH, and to ER, and then eject the palmitate and repeat. So we have megasyntheses of these assembled, which allow for the rapid synthesis and efficient synthesis of palmitic acid. And lastly, and briefly, I promise, it's important to mention that fatty acid, our C16 fatty acid, palmitate, does undergo frequently elongation and unsaturation. So, example, arachidonic acid has 24 carbons. It has a number of points of unsaturation, plenty of double bonds. It's essential for inflammatory processes and immune response. Lots of our fatty acids in our body are not 16 carbon, they're 18 or 20 or 24. And they're not all saturated. Many of them have 1, 2, or more double bonds. And so there's a number of biosynthetic pathways that check the boxes for elongation and unsaturation to create variations on palmitic acid. I'm not going to elaborate on those specific pathways. Just know that both of those things are very important in terms of the big scheme of things. But in the context of the remainder of this course, you need to be aware that it's a thing, not necessarily recite the details surrounding it. So with that, that's the final lecture for the semester. I'm going to get this uploaded for you, and I'll be talking to you all Monday in recitation.